The objective of this presentation is to gain insights into the motion of guest molecules inside DDR. DDR is an all-silica zeolite that has a cage window structure. This shows the structure of one unit cell that contains 12 cages. Each cage is 278 cubic angstrom. The cage volume is significantly smaller than for faugersite and ziphate. The cages are separated from one another by windows that are approximately 4 angstrom in diameter. This shows the dimensions of the 8-ring windows separating cages. The smallest distance is 3.65 angstrom and the longest distance is about 4.4 angstrom. Further structural details of DDR zeolite are provided in this table. These are the unit cell dimensions ABC, pore volume, surface area, to gain insights into molecular motions in DDR zeolite. We conducted molecular dynamic simulations at 300 Kelvin. Details of the simulation methodology and data on diffusivities are provided in the published literature. Let's have a look at CO2 diffusion. CO2 is a pencil-like molecule with a much smaller cross-sectional size, 3.1 angstrom, and a much longer length, 5.4 angstrom. The CO2 molecule can only diffuse lengthwise from one cage to the adjacent cage. In this slide, in this animation, the trajectory followed by a single CO2 molecule is followed as a function of time. You will note from these animations that CO2 spends a disproportionately large amount of time near the window and is preferentially perched at the window side. This has important consequences for separations, as we shall discuss in subsequent slides. Methane is much more strongly constrained at the window. It is able to cross the window because the windows are not completely rigid and have some degree of flexibility. Predominantly, the hopping of methane molecules in DDR is occurring within the cages. As you can see from the trajectory of a single methane molecule, it is located within the cages. Only very infrequently, once in a blue moon, is a methane molecule able to undergo a cage-to-cage hop. And the diffusivity of methane is significantly lower by about 2 orders of magnitude than the diffusivity of CO2. So let's look at CO2CH4 mixture diffusion. You can see the CO2 perched at the window and is able to cross the window in a lengthwise manner. Whereas in this for the duration of this animation, no methane molecule has crossed the window. Due to the strong absorption of CO2 and preferential location at the window sites, the CH4 diffusion is hindered by CO2. And the CO2 diffusivity is about 2 orders of magnitude higher than that of CH4. And therefore the CO2 methane selectivity in a membrane construct is significantly enhanced by diffusion, because both absorption and diffusion favor CO2. The company called NGK Insulators in Japan offer membrane modules which have a monolithic structure, and the inner walls of the tubes are coated with a thin layer, typically about 20 micrometers in size, this layer. And this layer consists of DDR zeolite that allows selective CO2 permeation from a CO2 methane mixture. Hydrogen CO2 mixture diffusion has a somewhat different character, because hydrogen is a much smaller molecule and can hop easily between cages. So the diffusion selectivity is in favor of hydrogen, but the absorption selectivity is significantly in favor of CO2. For a DDR membrane, the absorption selectivity in favor of CO2 overrides the diffusion selectivity that favors hydrogen, because of one important reason is that the hydrogen transfer between cages is hindered due to this preferential perching of CO2 at the window regions. In other words, even though hydrogen has a very high mobility and can hop frequently between cages on its own, the hopping, intercage hopping, is somewhat hindered by the preferential location of CO2 at the window sides. A DDR membrane is selective to CO2. Since DDR membranes can also be used to separate water alcohol mixture. To get an understanding of this, let's look at how water molecules diffuse. One important characteristic of water diffusion is the formation of molecular clusters. The molecular clusters also occur for methanol and ethanol. Due to clustering, a cluster of molecule has a lower mobility than either of the individual molecules that are unclustered. And this results in a mutual slowing down effect. And the understanding of slowing down effects caused by molecular clustering is essential to the development of DDR membranes for water alcohol separations.